Hey gang, I want to talk you through the genetic disorder project and some database searches for the project. So in this project, you're going to become an expert on a genetic disorder, and you're going to create some kind of artifact to teach others about your genetic disorder. Now, you're going to start off with a mutated DNA sequence. So I'm going to provide a DNA sequence, a mutated DNA sequence, and you're going to find out which gene that sequence is for and what disease that you're going to become an expert on. So I provide these sequences, so you'll get one of these sequences, and I'm going to show you how to take this sequence and figure out your genetic disorder. So I have my own sequence here. I'm going to copy it. Then we're going to go to the NCBI site, the BLAST site. BLAST is basically a technique that we use in which we have a sequence, and we're going to check a database of other sequences and figure out which sequences are most similar to our sequence. So in other words, we're going to take out your mutated sequence, you're going to search the database for that sequence, and theoretically what you should get back, the most similar sequence to your mutated sequence should be the original wild type sequence. And then you'll be able to identify the gene, the protein, and the disease. So I'm on the BLAST site, and what I'm going to do is I'm going to click on nucleotide BLAST. They're going to take my sequence, I'm going to paste it in, then I'm going to go down to BLAST, and it might take some time searching the database for all the sequences that are similar to this sequence. And so you can see that I have a bunch of hits. So these red marks show the different hits, the different sequences that are similar to my sequence. So you can see these on the top are very similar, and you can see some up here. The ones at the top are the most similar to it. Or I can just scroll down, and you see here it tells me the name of the different genes. So this is not a human, so I'm not interested in that. But look, it says here Homo sapiens. So I'm going to click on that. And now the name of the gene, you see it says gene for HBB, which is hemoglobin beta. So I know that my protein, my gene, and my protein is going to be for hemoglobin beta. And you can see down here it tells you how similar your sequence is to this hemoglobin beta. It looks like it's 100% identical. You see right here, 441 or 441, and it goes by query 1, subject 1. So the query is your sequence, and subject 1. This is what has been returned to you, and it goes up base by base. The lines mean that they're identical. If there were any gaps, any missing lines, that would show that there is a different base. So now what I can do is I can search information about hemoglobin beta, and then that should be one of the genetic disorders on the list. So if you look here, now it's not one of these diseases because I'm using a different sequence, but then you would do some research, maybe a Google search, or I shared some links with you for some research down here, and you can search some of these sites to see if you can find out which disease your protein or your gene is associated with. Now, once you have identified your disease, so I've already identified my disease as sickle cell anemia, and then I can find the other people in my group that are also researching sickle cell anemia, and then we can follow the research directions below. Now, one thing I should point out to you is that one person is responsible for writing the research for each of these topics. So you have 3 people in your group. And so, for example, if I am the author if I'm student one, then I'm the author for symptoms and treatment, but then I'm also the commenter for inheritance and epidemiology. So after student three has written up this segment, I am going to comment on that writing. So to do a comment, let's say this is some of the work. I notice there's an error here, or I have a suggestion. Go to insert, comment, and then I can type my comment, right? And then if I'm student one, I'm also an editor for this section. So I will find the comments in this section and resolve them. So let's just go back here for a second. So let's say there's a comment here. Well, I can, make my edits to the document and then go here and click resolve. And I will be able to find a list of all the comments there. So hopefully that was helpful. Now, in terms of the artifacts, your group it's going to make one artifact. You guys have to decide, would you like to make a poster about your genetic disorder? Would you like to record a video presentation, so similar to like a PowerPoint but recorded on video? Do you want to create a web page? Do you want to do some kind of creative skit, which would have to be recorded on video? And there's other formats that you might want to use. You can decide. Just clear it with me if it's not one of these that are on the list. So hopefully that was some helpful information. Thanks, guys.